Okay, so right now what we're going to do is we are going to compare different species based off of their DNA sequence. So this is the most molecular way that you can compare living things to each other. So I'm going to look over here at this data table, follow my cursor. I have an unknown species, and I have species 1, 2, 3, and 4. And I'm given a little snip of their DNA taken probably, actually for sure, from the same location. So let's see, let's compare their DNA and figure out how we can make some sort of tree or cladogram with this information. So this is going to be a generic way that you can organize data when you are looking at similarities or differences in DNA or amino acids. So I'm going to look at similarities. Differences will give you the same answer. So this will be similarities in DNA. You do not have to write this out. It will not be graded. In fact, if you have a scratch piece of paper, good, a computer paper, it would be actually very helpful for you to print this because you're going to have to compare the different species and it gets a little bit difficult viewing it on a screen. So I'm going to do unknown as UK, species 1, species 2, 3, and 4. And do the same across the top. Unknown, species 1, species 2, species 3, species 4. Okay, so I have two halves to this data table. I've got the top half and I've got the bottom half, and they would have the exact same number. So I'm only going to fill out half of this data table. Let me explain. Let's start, if you're following with me in lined paper, by drawing our grid lines. Not necessary, but I believe to be very helpful. Okay, so now we're going to make our dividing line by putting X's in boxes that don't make sense. You can't compare something with itself. So whenever you're comparing something with itself, by bringing in the side and the top, you put an X. So comparing unknown to unknown, that's nonsensical. Species 1 to 1, species 2 to 2, 3 to 3, you can't compare something with itself. So as you guys can see, I have created a divide. The top half and the bottom half. They would both be the same if I were to fill it out completely. In fact, in my research, I found that some people put similarities on the top and differences on the bottom, but we are not going to do that. So I'm just going to cross out the bottom. We do not need it. Okay, so now it's time to count. This is honestly kind of fun. So what I'm going to do is I'm going to compare the amount of similarities between species 1 and the unknown. It's like Punnett squares. You pull the side and the top together. Okay, so the more similarities they have, the more closely they are related in evolution. That is a key point for this lesson. So let's go over here, follow my cursor. Over here, I'm on unknown species and species 1. I'm just simply going to count of these 9 amino acids, how many of them are the same. 1, 2, 3, 4, 5, 6, 7. I record the number 7. Now I'm going to compare unknown to species 2. 1, 2, 3, 4, 5. 5. All right, now unknown to species 3. I'm just like mentally blocking these two middle boxes out. 1, 2, 3, 4. 4. Now the unknown to species 4. 1, 2, 3, 4, 5, 6, 7, 8. Wow, that is significant. Okay, so I can look at this and I can figure out who is the most closely related. So I have species 4 and the unknown have 8 similarities. So I can say that it is the most related to species 4. And I can say it is the least related to species 3. So I can make a cladogram like this. And I can put my unknown right here. This is who I'm comparing it to. And then it has the most similarities with species 4. It has the second most with species 1. The third most with species 2. And based off of their DNA, it is the least related to species 3. So you use the DNA evidence to create a cladogram. Sometimes when you can make one just with the top row, fine. Sometimes you'll have to fill out the additional rows down here. Okay, next, instead of comparing like we did over here, the DNA similarities between their ATCG code, we're going to look at the result of that, which is the amino acid sequence. So looking over here on the left, we are comparing humans to horses, gorillas, chimpanzees, and zebras based off of a 4 amino acid snippet of the hemoglobin molecule, which I believe is 15,000 amino acids. So this is titled Sequence of Amino Acids in the same part of the hemoglobin molecule. Just some background here. Hemoglobin is the protein that carries oxygen on your red blood cells. So if you were to zoom into a red blood cell, you would see, this is actually a pretty good picture, millions of these hemoglobin proteins, each with the capacity to carry 4 oxygens. So they just took this protein, stretched it out into its amino acid sequence, and did a snippet in the same place of 4 amino acids. And it's going to be the same concept. The more amino acids something shares, the more similar the species is. Well, you might be thinking they all have hemoglobin, so they're all related. Although that is true, there is some differences in their hemoglobin structure where you actually have to count the amino acids and the similarities and differences to see which ones are the most related based off of the nuances in their amino acid hemoglobin sequence. That is what we are going to do. So I'm going to go down here to the second half of this sheet. And I'm going to put in here similarities in amino acids. Okay, and I'm just going to put my species going down and across. So we have humans and horse, gorillas, chimps, zebras. And across here, humans, horse, gorilla, chimp, zebra. I'm going to make my guidelines here. I'm going to go ahead and get ready to count my similarities. Again, I could count differences. I would get the exact same thing. Okay, so I'm going to make my dividing line by putting an X through the boxes that don't make sense. You can't compare something to itself. I'm going to decide that I'm going to fill out the top part only. These bottom boxes would be the exact same thing. So we are now ready to count. If you're not and you're filling out the notes along with me, go ahead and press pause. But let's go over to the left side. Let's do our first comparison between the horse and the human. So I'm counting 1 similarity between the human and the gorilla. 1, 2, 3 similarities. Between the human and the chimp, 1, 2, 3, 4 similarities. And the human and the zebra, 1 similarity. Okay, so it looks like I can make parts of my cladogram right now and save some time by not filling out the boxes that I do not need. So I'm going to make a diagonal line. I'm going to put at the top what I know to be the top of this cladogram, which is the most advanced species on the planet, humans. If I didn't know that, I'd have to use some other data to figure that out, but humans always go at the top. All right, what is the most related to humans? We can put right here your chimp, because it has 4 similarities. And the second runner-up is the gorilla. But now we have horse and zebra coming in both with 1 similarity. So I need to do a comparison to get some more data with the gorilla. What's going to be next door to the gorilla is the question now. What's going to go here, and then what's going to go here? So let's go to the gorilla line. I'm going to fill out the zebra and the gorilla to the horse right here. So I'll start with horse, gorilla. I'm going to go over here. Oh, good. They're next to each other. 2. Now the gorilla to the zebra. Go, good, 1. So next door to the gorilla, we put the horse. And next to the horse, we put the zebra. We have made a logical cladogram based off of amino acid sequence similarities.